Hello everyone. Hello everyone. My name is Chao Deng. I'm a PhD student from the School of Computer Science and Engineering at the Central Source University. It is my great pleasure today to present our work, identifying new cancer genes based on the integration of annotated gene sets via hypergraph neural networks. First is the introduction of our study. In the last decade, large-scale cancer sequence projects such as the TCGA and the PCAWG have published genomic and transcriptomic data from tens of thousands of tumor samples. This large-scale cancer sample data have largely contributed to the development of cancer genomic research. A key goal in cancer genomic research is to identify cancer genes that play a crucial driving role in the development and the progression of tumors. Although the network of cancer genes and the cosmic cancer gene sensors have been used to annotate some cancer genes, the current catalog of no cancer genes is not incomplete. Accurate identification of cancer genes from many candidate genes remains a critical challenge. Currently, many computational methods have been developed to identify cancer genes. These methods can be roughly divided into three categories: mutation patterns-based methods, machine learning methods based on the handcrafted features, and the network-based methods. The network-based cancer gene prediction methods have been extensively studied in recent years. However, there are still some challenges with these methods. The cancer genes have been shown to cluster in a small number of biological processes, hallmark signaling pathways, and interacting subnetworks. But current research on the cancer gene prediction methods focus on the binary association of cancer gene in biological networks, while ignoring the functional association of cancer genes in annotated gene sets, such as biological process and the signaling pathways. The annotated gene sets encode a group of genes with functional association. In addition, cancer-driven mutation can lead to oncogenic properties of cell by altering the activities of hallmark biological process and pathways. Therefore, compared with the binary association in biological networks, the higher-order association information in annotated gene sets provides a more comprehensive characterization of functional association patterns of cancer genes and help us identify cancer genes more accurately. However, due to the complexity of higher-order association information in annotated gene sets, this information is difficult to use. So, although the knowledge of annotated gene sets is crucial for cancer gene prediction, using the knowledge to identify cancer genes face the following problems. How to represent the higher-order association among multiple genes in annotated gene sets. How to integrate different types of annotated gene sets, such as gene ontology, signaling pathways, and the human phenotype ontology, and how to make full use of the higher-order functional association information to identify cancer genes. To tackle these problems, we introduce this hyper. This hyper is a novel method to identify cancer genes based on the annotated gene sets and the hypergraph neural networks. The next part is ORS methods. First, we collect pan-cancer data to evaluate model performance for predict new cancer genes. Specifically, we collect non-cancer genes from the three cancer gene database as positive samples, and the negative samples are the remains genes after recursive including the non-cancer genes and the KGG cancer pathways gene sets. And we collect multiple types of annotated gene sets from the molecule signature database. We use C2 and C5 gene sets from the MSIGDB database, which contains signal pathways data from expert database such as Reactome and KGG, and the ontology gene sets from GO and HPO. This is the overall flow of our methods. On the next, I will introduce our methods in detail. Under the step 1, we try to solve the first problem. The hypergraph is an extension of the graph, and that can be used to represent the relationships among multiple objects. The hypergraph provides a natural way to represent the higher-order association relationships between multiple genes in annotated gene sets. So we represent the integrate different types of annotated gene sets using the hypergraph. And each gene is a node in the hypergraph, and each annotated gene set is a hyperedge. And we can easily integrate multiple types of annotated gene sets through a hypergraph. The step 2, we further solve the second problem. And in the node classification task of the hypergraph, the weight assigned to a hyperedge reflects its significance in the classification process. The weight matrix in the initial hypergraph is the identity matrix, which means each annotated gene set has the same importance for that cancer gene prediction task. However, the different annotated gene sets obviously have different degree of impact on the cancer. For example, the biological process associated with cell proliferation and the cell metastasis are more likely associated with cancer. Consider, Considering the association specificity of cancer to different annotated gene sets, we propose the disease-specific hyperedge weighting module. In this module, we use the non-cancer genes as the prior information for cancer and calculate the proportion of no-cancer genes in each annotated gene set as hyperedge weights. With this module, we can extend this hyper to more complex diseases, such as Alzheimer's disease and autism or Alzheimer's disease. In the step 3, we try to solve the third problem. The HGNN is a semi-supervised hypergraph node classification method and a powerful hypergraph learning method. HGNN extracts high-word association information in the hypergraph through the two-stage message parsing neural network between nodes and the hyperedges. So, staking multiple layers of HGNN have the risk of oversmoothing, which limits the expressiveness of HGNN and makes the model difficult to converge. In order to alleviate the oversmoothing problem of HGNN and enhance the expressiveness of the model, we propose hypergraph residual neural networks. We use this residual network approach to prevent the HGN model from being oversmoothed. We obtain the feature representation of genes through a three-layer hypergraph neural network and using a fully connected layer for final classification. The next part is results. The results are organized into six aspects, including performance analysis of this hyper and the analysis of prediction results. Firstly, we evaluate the performance of this hyper using five-fold cross-validation on the independent test size. We not only significantly outperform other advanced methods on the variance in five-fold cross-validation, but also achieve better performance on two independent test sites. This result shows that this hyper have stronger predictive and generalization abilities. We next perform ablation analysis on this hyper. The first result shows that integrate multiple types of annotated gene sets can help us identify cancer genes more accurately. And the results also illustrate that this hyper can effectively integrate knowledge from multiple types of annotated gene sets. The second result shows that the effectiveness of the hypergraph residual learning module and the disease-specific hyperage weighting module. Next, we analyze the prediction results of this hyper. We first use the cancer genes collected in the CancerMine database to evaluate our ranking of cancer genes. We assess the effectiveness of this hyper-cancer genes ranking by the distribution of annotated cancer genes in each decile of ranking results. The distribution of CancerMine annotated gene sets in this hyper-ranking results shows that the top-ranked genes in the prediction are more likely to be cancer genes. And so, we take this hyper-predicted top-ranked 200 genes as all predicted cancer genes and conduct further analysis of these genes. We then use the multiple independent functional genomic data to analyze PCG, including sleeping built-in transpose data, cancer gene fusion data, epigenetic regulator, and the PPI networks. PCGs have significantly enriched in this data and show the same characteristics as non-cancer genes. And next, we use functional validation experiment results to further evaluate these hyper prediction results. We use the results from the DepMap database to filter a group of essential genes that significantly affect the survival of multiple cancer cells in CRISPR loss-of-function experiments. We found that PCG have significant enrichment in essential genes. The results shows that this hyper not only effectively identifies genes that significantly affect the survival of multiple cancer cells, but also that the majority of PCG inhibit cancer-specific cell lethality. We also conduct enrichment analysis on PCG using the geo database and the two pathway database through the David. Enrichment analysis results show that all prediction results are significantly enriched in the cancer-related geo turn or pathways. Detailed results can be found in our manuscript and the supplement materials. And in PCG, 14-4 genes have neither in cancer or nor in NCG and the cosmic CGC. We consider these 14-4 genes as the novel cancer genes. To establish the potential role of novel cancer genes as cancer driver genes, we conducted a comprehensive analysis using cancer sample data from the TCGA study. We found that these 14-4 novel cancer genes have significantly differential expressed and differential methylated in a variety of cancers, and are also differentially expressed in the pathological diagnosis stage of cancer. And finally, we performed survival analysis of 14-4 novel cancer genes of multiple cancer types. We found that 14-3 of 14-4 novel cancer genes have significantly survival analysis results in multiple cancer types. This finding suggests that these 14-4 novel cancer genes may influence tumoral genesis and the progression in diverse ways. In the end, I want to thank my supervisor, Professor Jianxin Wang, Professor Hongdong Li in the Central Source University, and Professor Yaohan Li in the Old Dominion University. And lastly, I would like to express my sincere gratitude to ISCB for providing the travel fellowship. Okay, that's all. Thank you for your attention. Okay.
Thank you very much. We have maybe time for one or two quick questions.
Okay, I can start. I was wondering at the beginning when you select the known cancer genes as positive and all the remaining genes as negative, finally the negative set will contain the positive genes you want to discover, and you will have also a class imbalance. So how did you manage that?
Okay, let me think. Yes, the class imbalance. Yes, we have the 696 positive sample and the uh, approximate 200 negative sample genes. Although this class imbalance problem exist, but I think this distribution of the cancer genes can be extended to all the cancer genes. Maybe I can't express uh, my okay the problem. Maybe you can send me an email and we can detailed answer your question. Okay.
Sounds good. In terms of time, we're going to move on. Thank you again. There's the email address if you have questions.